Howdy everyone, you're listening to Blue Jeans and Boots, where genetics grad students talk about how to navigate a Ph.D. program while trying to stay sane. We also often invite faculty from our interdisciplinary program here at Texas A&M, or researchers outside of A&M, onto the show and pick their brains about their research, what it's like being a principal investigator, and their journey to becoming a professor here at A&M, or, in special cases like today's episode, at places other than A&M.
In this episode, we'll talk to Dr. Harmit Malik. Dr. Malik is a professor and associate director of the Basic Science Division at Fred Hutchinson Cancer Center in Seattle, Washington. He received his BTech in chemical engineering from the Indian Institute of Technology and his Ph.D. in biology at the University of Rochester. His lab studies the evolution and genetic conflict in Drosophila, primates, and yeast, with a focus on centromeres, viruses, and mobile elements.
My name is Emmarie, and I'll be one of your hosts today.
And my name is Veronika, and I'll be your other host today.
So, first things first, can you tell us a little bit about your research, or your current research?
Howdy, folks. Thanks for having me on today. So my lab is very interested in genetic conflicts, or what we colloquially refer to as red queen interactions. These conflicts occur when different genetic entities vie with each other for evolutionary dominance. The inspiration really comes from prey-predator interactions, where prey are constantly sort of fighting the adaptive strategies of predators, and predators are responding in order to maintain some sort of fitness equilibrium. We are very interested in how many other examples within genomes can we see of these kinds of prey-predator-like interactions. So it's very easy, for example, to make the stretch from a prey-predator interaction between that and an immune pathogen interaction, because that's very much like the same dynamics. But that is, of course, occurring between completely different genetic entities. We are also very interested in identifying these kinds of dynamics occurring between different entities in the same genome, where we have now different entities that are vying with each other for evolutionary dominance, often to the detriment of the host genome where all of these battles are playing out.
So you mentioned that it's a detriment. How is it a detriment?
So a very good example is that we do study, for instance, conflicts that occur between selfish genetic elements like jumping genes or transposons and the genome. Often the genome is trying to curb the proliferation of these transposable elements, because that's in the best interest of the genome, because the runaway expansion of transposable elements means that there's a very high likelihood that they will jump into a gene that is really important for the organism, or they can actually just perturb the fertility, potentially sterilizing the organism because of a runaway expansion that occurs. This is also sort of one of those things where there is a little bit of a ticking time bomb, because the more identical pieces of DNA you have in your genome, the more likely it is that you're going to undergo some sort of deleterious recombination because of those elements. So not only is there an immediate impact, which is deleterious, but there's also a growth that occurs with over time, where the proliferation increases the risk of genome instability. And we do see many cancers, for instance, that are seeded by recombination events that actually started off between two transposable elements that are on different chromosomes or different parts of the same chromosome.
Okay, so I guess I have a question in the sense that you mentioned that there can be significant functional consequences if these genes or jumping genes go into functionally active genes. Is there a selection for that happening, or is there a drive that's driving those genes to insert themselves in those regions?
Yeah, there's a little bit of controversy about that in terms of how people think about the adaptive benefit that transposons might provide. Over evolutionary time, we have many instances where a particular adaptation actually only occurred because this transposable element made it happen. But it's a little bit of the sort of benefit of hindsight kind of equation. In other words, there is adaptation that can be attributed to transposable elements, but is the net consequence of the transposable element beneficial or detrimental? I think the overwhelming answer is it is detrimental. It's a little bit like trying to paint some masterpiece, but you basically have like a shotgun, which is sort of what the transposable elements are. Now, there are exceptions to this. There are some transposable elements that have been domesticated or curbed enough so that we can direct their transposition properties just to some parts of the genome. A really excellent example of that is what happens in your immune cells, where you undergo VDJ recombination in specific parts of the genome, that is also actually derived from a transposable element's guts. And now the transposable element is not actually jumping itself, it's actually carrying out this transposition reaction. So you've effectively dismantled this transposon, and you've kept all of the elements of the transposon in order to mediate this completely orchestrated genome rearrangement, because you have complete control over when to turn on the gene, when to turn on the protein, what are the genetic elements. So those are the kinds of things that where we can see that the transposon is now benefiting. But there are actually only a handful of examples where actively jumping transposons have been shown to be unambiguously beneficial for the host. We can talk about what those examples are, because there are actually very few of them.
I'm actually curious about, you work with Drosophila primates and yeast. So I guess I'm curious about the benefits of each of these model organisms. Like, why do you focus on these ones?
Yeah, that's a very good question. I've often wondered that myself. I think part of the reason is it sort of slightly stems from what is the question that we are actually trying to answer. And then oftentimes people try to fit their question to whatever model system that they're actually studying or comfortable with. I sort of realized relatively early in my career that that's probably the wrong way to go about it. If you're really interested in the question, we should spend some time trying to figure out what is the best system to study that particular question. And even though it can take some time to get that system set up in the lab, you can often do so quite readily with the help of collaborations, etc. The added benefit is that whenever we worked on these additional systems, they're a little bit on a short-term basis. Usually they come in because there's a fantastic graduate student or a postdoc who's interested in a question. And especially if they're a postdoc who want to set up their own lab eventually, the advantage of them setting up that system in my lab is that when they leave, they will take everything with them. And so we have worked on a number of systems. We don't simultaneously work on all of the systems, but we do have now this sort of built-in memory about having worked on yeast, so we don't feel so intimidated about the fact that we can kind of go back to that. Some systems are a lot more intimidating to kind of venture into. We have projects that we're trying to kind of get off the ground in mosquitoes right now. That's not something that you do on a dilettante basis, as you know. We've had projects where we basically had a menagerie of different insect species, including we were growing silk moths for different reasons, and I had a bunch of Lepidoptera. That was really exciting and fun, but also completely terrifying because we really did not know what we were doing.
Is the main learning curve when you're going across these different model organisms is trying to maybe recruit or make sure that you have the right resources and knowledge base to start answering those questions? Because Drosophila and primates are really different.
Very different.
Well, just to clarify, we do not work on primates. We mostly work on primate cell lines. So that's a lot different.
Yes.
But we are trying to model a lot of primate evolution using cell lines in our work. And so in some sense, cell lines, whether they're Drosophila cell lines or whether they're microscopic organisms like yeast or bacteria, or primate cell lines largely are similar. I think the biggest kind of thing that we worry about a little bit is, are we just basically going to spend all of our time reinventing the wheel that other labs that are really very adept at this are going to do? And that is a worry that we have. That's not the goal of what we're doing. We're studying this because we're interested in a particular question, and we definitely don't want to spend all of our time reinventing the wheel. So what we do is we try to identify collaborators, often co-mentors for the trainees who are interested in the work, who are going to help them sort of accelerate through all of the growing pains of adapting to a new system. And if we cannot find a really good situation where that sort of technical hurdles cannot be overcome, we sometimes will move on from a project, because I definitely don't want to waste a lot of my trainees' time just trying to make them as good as many other labs that are already very good at that. And because, we want them to reap the benefits of that system in order to ask the question that they want. If they're going to spend 3 years just getting the system up and running, it's just not worth it.
Yeah, that's totally fair. Before, I do have a couple of questions about mentoring philosophy and I guess some of the traits that you would look for if you were recruiting or hiring a graduate student or a trainee. But I had some questions about fossil viruses and how they affect our genomes. So could you tell us a little bit about what fossil viruses are, and how can you determine their presence, I guess, in our genomes?
Yeah, so fossil viruses is a term that we use in a sort of colloquial sense, but many viruses, especially retroviruses, have an obligate step in their life cycle where they have to make an imprint in the host genome, and it is actually that imprint that is used to generate daughter viruses. If the cell survives the retrovirus infection, that cell will then be passed on. And if the cell happens to be a sperm or an egg-producing cell, the parent is not just passing on the infectious virus to their offspring, but they're also passing on the raw material for producing even more infectious viruses in the offspring. And so what we basically have here is a genetic legacy of a number of ancient retrovirus infections that made their way into the germline, which is the cells that produce sperm and egg, and have been passed on for millennia. At least, we can identify quite reliably retroviruses that date back about 35 to 50 million years. But like any other piece of the genome, if it's not under selection, it's going to be subject to mutation. And so that's where the fossil term comes in, because the longer they've been sitting in the genome, the more decrepit they get. And so we can actually date when they landed in the genome in two ways. We can date based on how many mutations they've accumulated, and we can date them by how many primates shared the exact same insertion in exact same location. So that actually provides us a very good sort of sanity check in terms of how old they are and how much of an impact they've had on the genome. It does actually surprise people when they learn, of course, if you're not in the business, you're surprised by some simple facts that we all take for granted. For instance, the human genome, you can recognizably sort of attribute 8% of the human genome to these fossil viruses, which in itself doesn't sound like a very impressive number, but less than 2% of the genome makes up all of the protein and RNA that the human genome finds. So, then you start thinking, okay, we have 4 times as much fossilized viruses, and yet there's a huge part of the genome that we haven't actually explained, and most of that has to do with selfish genetic elements like transposable elements we were talking about. And it's quite likely that the 54% that we don't really know what it is probably just, again, so fossilized that we can't even attribute which elements or viruses that they actually came from. So it's a little bit sobering for people to kind of realize that their genetic, imprint is really full of viruses and spam, so to speak. But that's just the nature of evolution.
That's so cool.
Can you describe any current fossil viruses that have affected our genome?
Well, the best example of a fossil virus that basically encodes a gene that is really important for us is a gene called syncytin or syncytin, which is actually derived from the envelope gene of a retrovirus that landed in our ancestors 35 million years ago. And while the rest of the retrovirus has completely abraded away because of mutation, the envelope gene, which is ironically the infectious causing gene of these retroviruses, has been basically retained in pristine condition. So much so that if you actually do an experiment where you take this gene and put it back into a retrovirus, replacing its envelope gene, it will act as an infectious agent. So, it's basically one of those very strange cases where the human or the primate genomes basically found this, piece of genetic gold, if you will, that landed into the genome because of an invasion and have basically decided to keep it around. And the reason they keep it around is because the cell that absolutely needs the properties associated with the retrovirus envelopes, both for cell fusion and for immune suppression, are the placenta. So it actually turns out that if you don't have syncytin gene function, you will not be able to carry pregnancies to term. So here's a really enormous example of something that we would associate as being perhaps one of the most fundamental things that a species can do, which is to biologically reproduce. But really the engineering for that mode of reproduction has basically come from a retrovirus.
Is that part, the fossil virus, present in all placental mammals? Have they identified it? Or they must have, right?
Syncytin gene the syncytin gene encoding imprint is only preserved in primates.
Okay.
But it actually turns out that every single lineage of placental mammals, except for marsupials, where the case is almost there, but not quite fully sealed. So cats and dogs, rabbits and hares, goats and sheep, and rodents, they each have their own syncytin that actually derive from their own private retrovirus, where the retroviruses are not even related, but they all have the same property of being expressed in the germline, and so they were able to invade the germline, can get retained, and now maintain this placental function. So if you were to infer what's going on is that we anticipate that when we made the leap from egg-laying reproduction, which is what birds and platypi do right now, into live birth, right, through placental reproduction, there was probably a retrovirus envelope gene at the heart of that. And what has been happening is that since retroviruses keep landing in our genome, it's like we get a choice every time a new retrovirus lands in the genome. Do we keep the old syncytin or do we trade up to a new syncytin? And apparently we've traded up several times over the course of mammalian evolution.
Okay, one last question about these fossil viruses. So do they tend to insert themselves, or I guess, in places of the genome that have lower recombination?
Not so much that they insert in places that have lower recombination, but we can actually identify them more readily if they're in places with low recombination. The reason is that most of these retroviruses, when they insert in the genome, are flanked by perfect long terminal repeats. And these repeats can undergo recombination that basically excises out the entire guts of the retrovirus. And if you're in a part of the genome that is highly recombinogenic, chances are that your full-length retroviral imprint is going to quickly be lost, and you'll only be left with a small nugget of an LTR. So it's not so much an insertional bias as it's an ascertainment bias in terms of recovery.
Okay, that makes sense.
Okay, well, moving on from retroviruses. So what influenced you to become interested in all of this with your background in chemical engineering?
Okay, this is a very interesting story, which actually has less to do with evolution of genes and genetic conflicts and more to do with my own evolution. Mm-hmm. So when I decided to major in chemical engineering, I was sort of a 16-year-old student. So we sort of made the determination prior to freshman year of college. And to be fair, I really did not know what chemical engineering was about. And even though I had some very good professors in chemical engineering, it was clear that I did not share the same passion for chemical engineering as many of my classmates did. And so I sort of started thinking about what I would do after, a degree in chemical engineering, mm-hmm. where I was pretty sure that was not going to be chemical engineering. So considered a number of options. And at the same time, there was a new professor who had actually started in a neighboring department, and he offered a course called Introduction to Molecular Biology, where he sort of went through the engineering principles of the lac operon, which is, one of the paradigms for these kinds of events. And I started attending these classes, and they were wonderful. He was a wonderful teacher. And then I realized that I couldn't continue because suddenly I was not attending the chemical engineering classes that I absolutely needed to graduate. And so I came to him and I sort of sheepishly said that I'm sorry, but I've been enjoying his classes. I just wanted him to know that I was not going to be able to attend. And I had been very active, kind of like asking questions. So he was a bit disappointed because he thought that, I was like clearly engaged in the material. And he said, well, why don't we do something? I'm going to teach these lectures in the morning. And if you want to come by in the afternoon, we can talk about what I covered. As long as you keep up with the reading, we can, you can sort of effectively audit this class. And I thought he was kidding, but he was not. I said, okay, I'll do that. And so he did that for a whole semester and then another semester. So he basically taught me, it was like this magical experience where he was teaching me molecular biology one-on-one. And at the time he was a chain smoker, so I was like literally taking like a pack of cigarettes as my penalty for like sitting across. But he was an absolutely amazing mentor, and it's actually really imprinted upon me the value of mentorship right from that moment, because I was not his student. I was not even in his department. He absolutely owed me nothing, and I just sort of was amazed. And now that I'm a professor and I sort of know how many demands there are on professors' time, it's just incredible that he spent, more than 100 hours just basically mentoring somebody who's not a student. So this is a little bit of an aside, but he was teaching me molecular biology, and I was reading The Selfish Gene by Richard Dawkins at the same time. And I began to recognize this paradox, which was that on the one hand, the cell is this orchestrated machine. People have likened it to like a Swiss watch-like mechanism. All the gears have to fit perfectly well together. On the other hand, we had this view of the cell where everyone was for themselves. The whole thing was going to, hell if you basically did not arrest these conflicts. And I became really excited about the fact that maybe there was a way to understand this paradox, and I thought that jumping genes would be like a great place to start. I'll just finish the story with my mentor. His name is Dr. Rao. He retired, and I couldn't make it to his retirement party, but I met him a few months later, and I kind of had to ask him. More than 30 years have elapsed since then, and I said, "Why did you do that? Why? I was kind of curious, right? It was like, okay, why would you take all that time to spend with the student?" And he smiled and he said, "Well, you just kept showing up." And, he told me he figured that I'm going to show up for a few days and then I'm going to stop because, it's not like really a class that I'm taking. Other pressures will come about. And he resolved, though, that as long as I kept coming, he was not going to be the one that would actually stop it. And that was like an incredible lesson, which is sometimes you just show up. You might not be having the best time and you might not be doing as successfully as you think you are, but actually just showing up is more than 50% of the battle. And the invigorating thing that you can do for a mentor by just showing up, even though you're not actually having the best time, is like actually really good because that invests in them to invest in you.
That was an amazing story.
I was going to say, I'm a little teary-eyed, not going to lie.
That was amazing. Thank you for sharing that.
I think it also speaks to so many levels too, because the mentorship, but also just I feel like both of us, the hosts, are early career, are starting early in grad school, first and second year. And it is so hard sometimes, but I feel like the passion is often what drives me to wake up and go to the lab. And it feels really nice to be reassured in a way that just showing up is it shows that you care and you're trying. So that I absolutely means a lot to me. Yes.
Well, there are lots of ups and downs in grad school, right? And as you go through, there are going to be, the thing that I always remind people is write down what got you excited. Why did you go down this path? It's actually important to remind yourself more than others, because sometimes we're in the throes of, your PCRs have failed again and your crosses are not taking because nobody likes, to mate with each other because your, fly suddenly decided they're asexual. All of those things happen. That's but actually, the thing is, if you just get lost in the minutiae of those things, you just sort of lose sight of why you're doing this in the first place. And honestly, the worst outcome would be that you graduate with all these skills, but you've lost the sort of enthusiasm and passion that brought you to grad school in the first place.
That's great advice. Actually, I have a whole bunch of, one of the reasons I am currently doing my current project is that I'm really interested in working at a museum, and I have actually a bunch of photos from the very first time I got to do a behind-the-scenes tour of the Field Museum in Chicago, which is originally where I'm from, and I got to see a bunch of specimens from species that are now currently extinct. And to me, one of the reasons I'm so passionate about conservation genetics is because I don't want to continue to see species go extinct, or at least preserve some of that biodiversity before it's lost. So having photos of that around my desk sort of reminds me, it's kind of morbid in a sense, right? Because they're they're extinct and they're no longer here, but it's a purpose. It's why I'm here. It's what I care about. It's what I want to do. So yeah, I think that's great.
I think it's a good inspiration. Actually, working in primate evolution, it's really staggering to know what, in my lifetime, what we have done to primates. And it's quite likely that in your lifetime, we will have some of our closest primate relatives will be extinct because of our activities. And that's sort of one of those things where just the lack of political will, the lack of appreciation for what we will lose. I mean, there are a lot of scientists who are well-meaning, who are big time into the de-extinction game and, like populating life on Mars and all of those stuffs. And yet we sort of have lost sight of what we had and what we still have, which is, an irreversible thing once we lose it.
Yeah, exactly. On a I don't know. A slightly different note, imposter syndrome is also really hard to deal with the reality of it. And prior to doing the interview, we had dinner, and you had mentioned that you had some pretty good advice, possibly, for dealing with imposter syndrome.
Well, I have advice. The good part is I think the first myth is that you will grow out of imposter syndrome. Mm-hmm. You will not.
Great.
And I think it's actually good for people to, you're sort of, you sit in a room and you're surrounded by brilliant people all the time. Mm-hmm. And it's actually okay to sort of feel a little bit intimidated. I mean, that's the point, is like to recognize that, there's some brilliant people in science. And one of the reasons we want to be in this field is to be surrounded by this brilliance and stuff but the thing, what you don't know is that they think the same about you, but they don't necessarily, they're not going to come out and say it unless they're very much into the self-affirmation type game. So, I've been a faculty member for 21 years, and I sit in rooms sometimes with some colleagues, and I'm just amazed by them, right? And they're not even senior colleagues. They're sometimes even junior colleagues. I was like, I cannot believe how amazing this person is. I just sat in a qualifying exam recently where, despite my very, very best efforts and trying to ruffle the feathers of this young person taking their qualifying exam, they were just absolutely rock solid. So when they left the room, all of the other faculty on the qualifying exam turned to me, because clearly we were having a back and forth. And I just said, I tried my best, but I could not ruffle it. And that is amazing, right? Mm-hmm. Like that is like truly amazing. Like, you get these examples. I think the bigger lesson, though, is, we look for inspiration in others, right? And it's actually totally normal. We got into this business because we were inspired by somebody, and somebody took a chance on us. But often the people that we are inspired are several career stages ahead of you. Yes. And that can be really intimidating when you think, like, how am I ever going to get there? They're like, they're in the National Academy. Some of them are Nobel laureates. They really have this absolute fantastic career, but they didn't get there overnight. And so the best advice I would give is, don't necessarily think of them as a path that you have to take, because that path has several steps that you cannot see. But the path that you can take is find somebody who, in your opinion, is killing it. That's just one career stage ahead of you. If you're a first year or second year student, find a fourth or fifth year student. Think about like what made them tick. Like, how did they succeed? What are they thinking? Are they going to do a postdoc? How are they going about that? Mm-hmm. Actually, that kind of practical advice is true even when you're a junior faculty member. One of my absolute best mentors and role models is actually my current boss. So I certainly hope she's listening to this. She's pretty amazing. But actually, for me, just following some of the things that she has done, in setting up her lab and setting up her program, has been in a way very freeing, because I'm not really trying to emulate somebody who's 30 years beyond my level. I'm just trying to emulate somebody who's 3 years beyond my level. And that's a much more achievable goal. And my piece of advice is, like with every problem you'll face scientifically, you have to kind of break it down, because when you add up all of the steps needed to solve the problem, they can be completely paralyzing. But actually, the first step is not so hard. And actually, all you have to do is just take it one step at a time. And that's the same advice for role models.
For you, as a current professor, what are some challenges that you have faced, and how did you overcome them?
I think the biggest thing that I think students should get about their professor, perhaps, is they miss being in the trenches. Mm-hmm. And it's actually kind of really hard to not be in the trenches, because that's kind of why we got into science to begin with, right? But if, as my lab manager puts it, like, if you're really interested in this project, give it to somebody else, because your bench is where projects go to die. Because, the thing is, there's so many other demands on your time. I don't even teach that much. I'm not at a university. I'm at a research center, etc. But there's still a lot of demands. There's other people's projects, and you need to put your thinking hat on for a number of projects. And I love doing that. But I also sometimes just love having my own puzzle to solve. And so sometimes when faculty members might be a little bit too pushy, I think it's not so much that they're trying to push you. I think they're just a little bit sad and frustrated that they have to live vicariously through their students. And that can be hard for a lot of people. And certainly there are times when it has been very hard for me, where I knew that the success of this project was not actually dependent on me. It was actually going to be completely dependent on the student. And the best I could do was to support the student so that they would feel empowered to succeed. But I had no, it's a little bit of a weird situation to be in. Now, the student might feel the same way. They'd be like, well, suddenly this professor is extra attentive to my project, and I wish they would leave me alone a little bit. But I think you can see that the tension is you just need to do it in a way that you don't intimidate the student. But there's a reason you're interested in science to begin with, right? And now you're not actually the one doing the science.
It's always funny. My advisor always tells us that one of the happiest times of his life ever was being a graduate student, and it wasn't because it wasn't difficult, because it was super, super hard, but the fact that he got to be intellectually curious and execute all these projects and get to talk about science all the time, it was great. He also, I think one of the things that I really respect about my advisor too is he's the type of person or professor where every success for me is also a success for him. I'll go run down to his office and be like, Bill, look what I just discovered. And he's like, oh my God, great job. And we'll have a whole conversation about it. So for people, I guess, who are interested in possibly going to grad school, one of the pieces of advice that I would suggest that you maybe consider is that try to find a PI that really supports you, that you feel comfortable going to when you do have successes, but also when you're teary-eyed and not everything is failing and you just need to be told that it's going to work out. Find a PI who will help you through those good and bad moments. But yeah,
I absolutely agree.
One of the best pieces of advice I got when I started my lab, I went around to talk to faculty colleagues who'd been doing this for a long time and asked them, so what should I know? I mean, I'm going to do this, for real. And they said, make sure you get Kleenex and put it in your office.
Wow.
And I said, was that for me or was that for my students? It'll be for both. But just make sure that when you're doing the crying that you close the door. And so, I mean, it's a very emotional thing to do what we do, right?
Yeah.
It's not the kind of thing, unfortunately or fortunately, when you go home, it's hard to switch off, especially when you're in grad school. You're thinking about how the day was. If you had an exciting result, you can't wait to get back into the lab. Sometimes you don't even leave the lab. And if it was a terrible day, very disappointing, it's also sort of really hard to let go of that. And the thing is, we have to kind of find a way to kind of process that so it doesn't completely take over. But it's also the kind of thing that, yes, make sure that you're in an environment, whether it's the advisor, whether it's other people, hopefully it's all of them, that just share the journey with you, because otherwise it can be kind of a little lonely.
Definitely. I guess with that in mind, are there any things that you like to do in your free time? Whether it was during grad school or currently, any hobbies that might surprise people listening in?
Some hidden secret talent.
Yeah.
There is the word talent kind of gives it away. There's not much talent, unfortunately. But I did actually, I used to be an avid reader, and I sort of began to rationalize that I really couldn't afford to spend that much time reading when, I have so many unread papers and theses and stuff. But actually, when the pandemic hit and, there was just a lot of forced free time, I did actually take up reading again. I really did enjoy it. I read, I think, 60 plus books in 2020. And I just sort of really enjoyed the rhythm of that. Now, I don't quite read that much anymore, but at least that muscle memory is still there. And I kind of feel like there are times when I do enjoy reading. I have a lot of graphic novels and comic books that I started purchasing when I was a graduate student. I think pretty much all my disposable income went into purchasing comic books. And I was sort of had this dream that I would eventually sell them and help pay for college for my kids, etc., I actually don't think they're worth very much. And I'm not sure that my wife really enjoys my hobby as much as I do. But once in a while, I will actually go back and I will read something. And it's actually kind of one of those things where it takes you back to when you were a grad student, when you first read that. Or, just like any other memory, it's sort of, and it's, it's nice to have that. But I, unfortunately, am not as talented as many people. So I mostly just try to identify talented people to work in my lab.
Interesting. I guess on the topic of books, are there any books that you would recommend that are sciencey? Out of those 60 books, were most of those sciencey or no, not at all?
There were not that many sciencey books. I would, since this is being recorded, perhaps I shouldn't say that I had a very strong affection to Scandinavian murder mysteries.
That is specific.
Very specific. But, I just went through an entire series of like Wallander. I read all the Wallanders back to back. And then it was kind of interesting. It's just like the mood setting, especially in like Sweden and Denmark, was very similar to like winters in Seattle. So it was like very evocative of like the kinds of things that, you have. But science books are kind of wonderful. I sort of actually think about science books as both a hobby as well as work, because I am actually inspired by them. And I actually think, my own career started with a science book that somebody wrote. And I actually think that a well-written science book is probably going to have a bigger impact than most scientists' career, because of the sheer sort of curiosity that they evoke, even in people who've been doing science for a long time. So I do have I do enjoy reading Carl Zimmer. I do enjoy reading Ed Yong. And I also sort of really enjoy some of my colleagues who've taken to like writing books themselves. Some of them have been more successful than others. And by success, I don't mean like they're on the New York bestseller, although some of them are, but they're just better at being able to evoke that sense of joy and curiosity.
Do you think that you would ever, or is there any desire for you to write a book?
Well, I have actually thought about it a lot. And I also, this is embarrassing to admit, I actually have 2 chapters of a hypothetical book already written, which I wrote sort of in a huff about paleovirology. But, I've realized that there is a lot of discipline that is needed to write a book, much more so than to write a paper. And I'm sort of reaching the sort of realization that I'm going to need somebody to drag me over the finish line if I'm going to do that. I'm actually very good at like a short-term sprint, but the books are more like a marathon.
Yeah,
absolutely.
And you can see me at the 1 mile marker, sort of huffing and puffing and not going to make it all the way. Yeah.
Would you compare it then to like writing a dissertation where you're like trying to put it all together and you can see the finish line, but you're like, oh man.
I think writing a dissertation is slightly different because you've already kind of run the race.
Okay, that's fair.
And you're simply sort of trying to splice together the video reel for the race. And I think the best way to think about the dissertation is that every single chapter you write, every paper you write, you're basically writing your dissertation. And it's actually really good to think about it that way, because by the time you're done, hopefully all you have to do is like put together an introduction and a discussion chapter. It does feel like a marathon though.
I'm curious about, what does a typical day look like for you in the lab?
A typical day for me in the lab is mostly involved with meetings with my trainees. And we are often either trying to put together a story for a paper, or we are basically mulling over puzzling results where we sort of try to draw and redraw models over and over again to try to figure out what's actually going on. It is sort of segmented though, in the sense that I usually devote an hour per meeting, and I don't necessarily meet all the people in my lab on the same day. So part of the job is also just to make sure that I check up on everybody in the course of a week, especially if I've been traveling like this week. When I get back into the lab, my first task would be to make sure that I just check up on everybody to make sure if they need me for something, or even if they don't think that they need me for something, we'll kind of go through. Some of this is just to make sure that things don't sort of become too big to then control in the end, and people don't waste a lot of time with something that, with advice I could have given them early on in the business. Both parts of the projects are really exciting. When people are getting started, like establishing a project, it's really exciting because there's a lot of potential, and I can sort of see the potential. They can see the potential. It's also sort of actually exciting sometimes because you're sort of stuck, but you know you're doing all the right things. And so some of it is just the fact that I've been in those situations before and they haven't. And so I'm just trying to like walk them through that. Like, this is a puzzle. And even though this puzzle could make the difference for your career, it is still a puzzle and you're really good at this. And sometimes it's just like reassuring them to just think through each of the steps. And then there's that moment of discovery, which unfortunately is not as, common as television shows would like you to believe. And then you really celebrate those because there's that moment where you realize that you have definitely figured something very cool out.
Yeah. Do you do any of the wet lab work as well, or it's all your students?
My lab does not even let me do mini preps anymore. I don't know whether you've ever done mini preps. Right now you can actually do mini preps with the kit. When I started in the business, there were no kits. That is how old I am. But they won't even let me do a kit because they just... I think the last time I actually offered to help a graduate student who was under time pressure to do some experiments in response to reviewers. And I said, you have to do these 64 mini preps. Why don't you do those 32 and I'll take the other 32? And her reaction was very much like a mother bear protecting her young. And a look of horror just said everything that I needed to hear. So I have not made that many offers since then.
Okay.
It is kind of a motor memory, though. I mean, once you're off the bench, I can still set up fly crosses pretty, confidently, but there are a lot of things where I would basically spend hours of other people's time just trying to figure out where things are.
Yeah. Yeah.
With that in mind, do you miss it? Like the wet lab work? I guess going back to kind of what you alluded to earlier.
I'm not sure whether I miss the wet lab work per se, but I sort of miss the fact that I don't get to do the sort of puzzle solving by myself, which was like very fun time to be on the bench and trying to figure stuff out. It was on the bench when I was a graduate student and a postdoc, large number of my discoveries were made on the computer. And even that I could do in my office, but there's just not the quantum of time. I think the biggest rate limiting sort of thing here is just attention span. So I do try to save attention span when I'm reading in a new field or etc. But even then, the priority is always to the people who are going to knock on your door with some exciting data, like you just said.
Yeah. Through the course of your career, what would you say has been one of the most surprising discoveries that either you or someone else in your lab has found?
I'd say that there have been a couple. At every stage, there's been something that is sort of really kind of driven. When I was a graduate student, the discovery we made was that there was a class of transposable elements called non-LTR retrotransposons, which are like very abundant in the human genome, that unlike what people knew about transposable elements, that is, they jump between species all over the place, we realized that actually many of these non-LTR retrotransposons actually did not jump at all or very rarely. And in fact, we could date them all the way back to nearly close to the origin of eukaryotes. So really, basically, they had been hitchhiking as, parts of eukaryotic genomes for more than 1 billion years. That was kind of surprising. It wasn't like it was a sort of a painstaking thing to get there, but it was actually quite exciting. But the one thing which was really an aha moment was using sort of a purely bioinformatic analysis, I sort of figured out and predicted where an enzymatic domain of a non-LTR element, which, other members of my lab were very interested in identifying and biochemically sort of characterizing. And I actually realized that they were looking in the wrong domain, purely based on, and, this was very early days, BLAST, etc., was like barely in its infancy. And I sort of realized that just by doing kind of multiple sequence alignments, and realized that this endonuclease domain was actually in the C-terminal part of the protein. And this person had spent more than 2 years characterizing the N-terminal part of the protein for good reason, because that's where other non-LTR elements had their endonuclease domain. But not only that, I actually realized that the non-LTR domain I had identified was probably related to restriction endonucleases, based on homology. And it was like one of those moments where I did the alignment, I dropped it off at my advisor's desk, and I didn't even have to say anything. Like he knew exactly, like he knew that it was it. And then, it was a little bit uncomfortable for the student who had spent the time. But on the other hand, I sort of, in a way, like greatly rescued her PhD thesis. And then within 3 months, we already had a paper. I was second author because obviously the biochemical characterization was most of the work. But it was very, very nice because it sort of helped my thesis also, because I could now fully analyze how these elements came into their configuration. And it's been nice because it's actually been, when I was a postdoc, I came up with this model for why centromeres are rapidly evolving. And that was awesome, except, I was very timid and my advisor was like, this has got to be right. And, we had a joke in the lab where every time he says this explains everything, it probably means we were like horribly off track. But actually in this case, we did explain everything, which was actually quite nice. And then in my own lab, there have been a couple of discoveries, but I think the one that most people know us for, which is what I'm going to actually talk about tomorrow, is our discovery that we can use signatures of rapid evolution rather than conservation to actually predict what host virus specificity domains are, down to amino acid precision. And, molecular biologists love to focus on evolutionarily conserved regions and evolutionarily conserved residues. And what we realized was that by focusing on the most rapidly evolving regions, we could actually figure out what the specificity domains were.
I was going to say, as a phylogeneticist, we love evolutionarily conserved regions of the genome. But yeah, that makes sense. That checks out. That's really cool, though.
What advice would you give to a student interested in pursuing a career in genetics?
I think the biggest advice I would give at the PhD level is just grow your skill set, build a strong foundation. I mean, it of course is important for you to have a successful thesis, but even just like having a strong foundation, good habits, those are good things. They may not be so perceptible as, for instance, numbers of papers or speaking engagements, etc. But building a good foundation in grad school is really important. And then when you're ready to make the transition and you want to do a postdoc, don't just think about where are you going to do a postdoc. Think about what is it that you ultimately want to do. The best advice when I interview people, I'm always asking them, what is this a springboard towards? If you're interviewing for a technician job in my lab, I want to know what are you going to do next. If you're going to interview for grad school in my lab, I want to know what you're going to do next. It doesn't matter whether you want to do academics or not. I just am interested in what you want to do so that I can help you with the transition. And similarly, if your ultimate goal is to run a lab or be a curator in a museum, it's really important to think of that and not just think of this as a stopgap measure until you get there. Everything is like, in a way, linked and a chain. And if you do decide to run your lab, think about what makes you unique. I mean, a lot of people are very attracted to areas that are really hot because they see a lot of papers in high-profile journals, etc. But the problem is that also just attracts a lot of competition, and it's really hard to kind of distinguish yourself from that. And I think what I have done in my career is mostly try to find the path less trodden. And that's been pretty successful.
Today's episode was hosted by myself, Emmarie Alexander,
and Veronika Mojic,
and was produced by Aaron DeSalvio. This podcast is brought to you by the Texas A&M University Interdisciplinary Graduate Program in Genetics and Genomics. If you have any questions about our program here at A&M, please feel free to email us at bluejeansandboots@gmail.com. You can also find us on Facebook, Instagram, and X at Genetics Podcast. Thank you for listening, and we hope that you'll tune in next time.